talking about diseases. In the last class, we were talking about sericulture morphology, that is silkworm morphology. In this class, we'll be talking about diseases and pests of mulberry silkworm, because any living organism will have different pests or diseases that will be occurring, because any living, the parasites need some living organism to survive on. So here, when these larval forms are so vulnerable, you can definitely expect different types of diseases affecting them. And of course, other than the diseases, there might be some pests. When you're calling pests, it can be any insects, which comprises of the arthropod insects. Yes. So here we will be talking a little bit of what are the diseases that are going to affect them, the pests that are going to kill these larval forms, and of course, the maintenance and the transportation of the mulberry silk, how we are going to do, and what are the next step after culturing the silk worms for silk threads. Okay, so coming to the first one over here, we'll be talking about the pests. We will talk in detail about the diseases, but what are the pests of silkworm? Any insect or a non-insect organism which interferes with the human welfare leading to economic loss is called as pest. Economic loss means when you're talking about some business point of view, when you're going at a loss, then you call them as economic loss, because culturing the silkworm means we are having them as a business. It can be a small scale industry, it can be a large scale industry, and when you're going to have your input so much, if you're going to have a loss, then that becomes an economic loss. And this economic loss is when it is brought about by any insect or non-insect organism. You call them as pest. Two important pests are found to cause economic loss. What are they? First thing is tachinid fly is one which comes under, it's a common name is oozy fly, what you call them. The scientific name is tachinid fly. Exorista bombyces is the scientific name of it, which attacks the larval stages. So common name of this tachinid fly is also called as oozy fly. This causes a decline in the cocoon yield. So if there is a decline in a cocoon yield, we are not getting silk. So definitely there is a loss over there. And sometimes in the cocoon stage, the silkworms are attacked by dermestid beetles. Beetles, they also come under arthropod phylum. Dermestid beetles or dermestid species. These beetles are commonly referred to as carpet beetles. They are reported to cause a considerable reduction in egg production in silkworm. Egg production centers or grainages where the silkworm eggs are produced, that is seed production is produced over there. They have a problem of this dermestid beetles. So under pests, we'll be talking about two, that is the larval, the oozy fly and dermestid beetles. Under diseases, we'll be talking about 4 diseases caused by protozoans, the virus, the bacteria, and the fungal. These are going to cause the protozoan diseases, the viral disease, bacterial and fungal disease. So we'll talk about it one by one. What are the causative agents, occurrence, and source of infection, and then prevention and how you're going to eradicate them. Okay. First one is a protozoan diseases, protozoan, phylum Protozoa, which are very, very microscopic. So the protozoan diseases is other name is pebrine disease, is the one caused by this protozoans. The causative agent is Nosema bombyces. This is the causative, the protozoan that we are talking about over here. The different strains of Microsporida is caused by them. Microsporida, micro means small, sporida means spore shape. So that is the strain which belongs to this Nosema bombyces. Non-seasonal, they don't have any season. Anytime they can occur and anytime they can cause this particular disease. Sources of infection. How is the silkworm going to get affected by this infection is through eggs or by eating contaminated mulberry leaf. So any contaminated mulberry leaf eaten by the silkworm, that is a larval forms, they are going to be infected by this protozoan disease. Sometimes infected silkworm, fecal matter of the larval forms, contamination of the rearing house, because what do you mean by rearing house? Where you rear or where you culture the larval form is called a rearing house. So if that is again contaminated, or appliances is here, you'll be talking about the cutting instruments, the scissors, the knives, the baskets that you're using, the stands where you keep these larval forms, the different appliances, it can be contaminated through that. And alternate host is also a problem over there. That is a mulberry pest, which are the source of infection. Mulberry pest means if the pests are infected by this disease, protozoan disease, then this is also going to cause this particular protozoan disease that is called pebrine. Nosema bombycis is a causative agent. So here you can see how you identify them, that particular larval form or the silk moth is affected by this pebrine is, if you can see the egg stage, when the egg has been laid by the moth, they are not evenly laid. You can see some clumps over there. So these clumps are one of the character of pebrine infected ones. When compared to the normal eggs, you can see the normal egg over here, they are laid very neatly one beside the other, but here you can see the clump formation because of the infection. And here in the larval forms, you have different sizes of the larva. They are not equal, because at any given instar larva, the larval size should be the same. But here, because of the infection, the growth of the larval form is affected. And this is the moth that has been infected by this protozoan ones. So you can see the wings have been diminished. They don't have a proper antennal formation. You can see the abdomen being completely infected over there. And this is the Nosema bombycis. This is the spores, microsporidia, what you call them. Under the microscope, if you look at them under the microscope, all these small pores are the Nosema bombycis. And how do you bring down the infection? By burning all the silkworm eggs, the moth and the larval form has to be burnt out, so that the infection is stopped then and there. Symptoms. So if pebrine is been affected, what are the symptoms the larval form show or the moth show? Irregular hatching of the silkworm, that is what we saw here. So you can find the irregular hatching of the eggs over there. Irregular size of the larval bodies and molting. The infected larva loses its appetite, becomes inactive with wrinkled skin. So you can see some wrinkled skin over there. Black pepper-like spots. Can you see those black pepper spots on the larval form? So these are the pepper spots on the body of the infected worms. And white pustules appear. White pustules, some pus-like formation appears on the silk gland, the internal gland. The silk gland, if you remember, yesterday we were talking about the silk gland. The silk gland is affected by this particular pebrine when examined under the microscope in presence of the shining oval spores, that is Nosema. Management, how you're going to manage, because always it is always prevention is better than cure. So how you're going to manage them? Disinfect the rearing house, conduct strict mother moth examination. You're going to check the moth. The female moth has to be checked regularly so that they don't lay any infected eggs, which will again increase the problem over there. And what you're going to do is you're going to surface disinfect of the silkworm eggs to produce and rear disease-free laying of the eggs. You have to follow strict hygienic maintenance during rearing. Control mulberry pests in and around the mulberry garden. That is, all those arthropod insects have to be controlled by spraying proper insecticides or pesticides. And you're going to apply bed disinfectant. Bed disinfectant is where the larval forms are reared in those beds, where you add those leaves on the beds. The beds have to be cleaned now and then, that is disinfected, so that there is no pebrine, that is Nosema, staying on the walls, which will later on infect the larva. Going to monitor the seed crops constantly to eliminate any microsporidia infection. So that was about pebrine. Coming to the viral disease, we will be talking one or two of each diseases over here. After that, protozoan disease, we'll be talking about this viral disease called Flachery disease. Flachery disease can be caused both by virus and bacteria over here. But here in this topic, in viral disease, we are going to talk about viral Flachery causative agent. Bombyx mori infections is caused by Flachery virus, which is also called as densonucleosis virus. Is the one that is caused by this particular Flachery diseases. It's common during summer and rainy season, very high hot season, that is the summer season, or the rainy season, both where the humidity ranges from extremes to low level over there. Sources of infection: silkworm gets infected by eating contaminated mulberry leaf. So always we are going to make sure that we have a proper mulberry leaf, which is not infected by any kind of parasites over there. Dead diseased silkworm, that is again a source of infection over there. Fecal matter, gut juices, which is secreted outside. The body fluids are also the source of pathogen contamination because it's a virus. It can easily come out of the body fluids. The infection can also take place through injuries, cut and wounds in case while handling. That is, when we are removing one instar larva and putting it into another bed, during handling, sometimes we might be a little bit hard. They might get some wound or cut, and the open space is a open invitation for any diseases to be infected. So especially the virus. Predisposing factors: fluctuation in temperature, high humidity. That explains why the occurrence is always during summer and the rainy season. And poor quality of leaf. That is, if the leaves are not of if not of good quality, if it is of infected ones, then definitely the larval forms are going to have this Flachery. So this is how they are going to look like. The gut extrusion, that is the secretions that are found from the larva. Here you can see how they are being dead, or the secretions that are coming out from the gut. So this is how the infected larva looks like when infected by this Flachery. Okay, what are the symptoms? The body becomes soft and flaccid. That's what we saw. Growth of the infected larva is retarded. They never grow properly, become inactive, and they vomit juice, gut juice. So this is what we saw. They are vomiting the gut juice outside. The feces becomes soft with high moisture content, sometimes chain type excreta, and rectal protrusions also are observed. Larval head and thorax become translucent because there is nothing left over. Everything is eaten away by this parasite. So they become translucent or then when infected, symptoms of toxicity such as paralysis and sudden death are observed in viral infected ones. After that, the death larva turns black in color and they give out a foul smell because of the virus that has been present inside. And sometimes dead larvae turn red when infected with Serratia species. So there are different types of species that are present. How you're going to manage? Of course, the first thing is always you're going to disinfect, that is prevention. The surrounding and also the equipments are being disinfected. We're going to provide good quality leaf, avoid starvation, because once they are starved, the immune system also comes down, so they are unable to survive. Overcrowding and accumulation also should be avoided, because overcrowding means you are giving less amount of space for them to move. And when they are in close contact, even if one larva is infected, with a close contact, they are infecting, the infection proceeds to another one. So that is the reason why they said during the coronavirus, overcrowding was avoided. And of course, we had to wear the mask, because that is how we are going to prevent the infection. Here, it is in our hands, so our overcrowding has to be avoided, and accumulation of feces in the rearing bed have to be cleaned up now and then, so that from the feces also this virus can be contaminating the other larval forms. You're going to rear the silkworms under optimum temperature and humidity. So you're going to maintain an optimum temperature and humidity also to be maintained, because high variations is going to lead to this viral infection. Coming to the fungal disease, or the name is muscardine. Till now we saw pebrine, Flachery. Now we are going to talk about the white muscardine disease caused by the fungal infection, fungus. Causative agent over here is the Beauveria bassiana is the name of the fungal, which is also called as white muscarine disease caused by this Beauveria bassiana. So you can see over here the larval forms over here having that cotton-like structures that is formed on the larval body. So that is the fungus. The fungus, you can usually see them on the bread. Whenever you leave the bread, you can see some hyphae growing on them. That is the fungus, the cotton-like structures growing on them. So the same thing over here. Fungus means anything that is having those cotton-like structures growing on them. Disease is common during rainy and winter season because fungus means definitely there should be a lot of moisture. Wherever there is moisture, you will definitely find this fungus to grow on them. Sources of infection. When infection starts when conidia come in contact with the silkworm, the conidia, that is the propagative part of the fungus over there. When they are coming in contact with the silkworm body, definitely the silkworm is affected. And mummified silkworm, mummified means what? They become very rigid, hard and rigid, mummified. Alternate host is maybe Lepidopteran pests. Contaminated rearing house and appliances are sources of infection. Predisposition factors, low temperature and high humidity is again another cause. That is rainy and winter, usually you have low temperature and high humidity. This is how the mummified larvae looks like over here, something like a chalk-like structures, hard and stifle kind of larval forms you can find. Here you can find during the cocoon formation also, they are being affected and this larvae are not able to produce a proper cocoon. And of course, you can see this one as a green muscardine disease. If this is a white muscardine disease, then this is a, because it's green in color, so you have that green muscardine disease. Why the color difference is because it depends on the type of the color that fungus is. If it is a white fungus, then it's white muscardine disease. And if it is a green color, you get a green muscardine disease silkworm. Okay, what's the symptoms? Loss of appetite, they become inactive. Moist specks are on the skin, moist specks. That is, if there's something watery like moisture present on the skin, also predisposes them to this fungal infection. Larva vomits and turns flaccid. They become very thin. After death, larva gradually becomes hard, followed by mummification due to the growth of aerial mycelia or conidia over the body, and they turn chalky white. So this is what they look like, a chalky white color. Management. I'm not going to talk about the management of the first one because you all know that rearing, that is a precaution, is important. We'll go to the next one. Pick up the diseased larvae before mummification and dispose them by burning, because burning is very much important. If you're going to burrow them or if you're going to throw them here and there, the conidia can move from there through aerial movement, and the chances of other larvae being infected. So definitely you will have to burn them. Avoid low temperature, high humidity in the rearing house. Relative bed humidity during rainy season by dusting slaked. You are going to regulate the humidity during rainy season by dusting slaked lime powder during mold. That is, when you're going to spray the slaked lime powder, what happens is you're going to remove a little bit of moisture. The moisture is removed, humidity is controlled over there. Thereby, you're reducing the fungal infection during molting period of the larva. Remember, the larva has to molt 4 times to get into 5 instar larva stages. So each molting, the larva is going to grow in bigger size. Okay, so that is called as molting. The next is the bacterial disease. I told you again, the Flacheae can be caused by the fungal, or also it can be caused by bacterial disease also. Okay. So what is the bacterial disease over here? Causative agent over here? Streptococcus, Staphylococcus, Serratia marcescens, and Bacillus thuringiensis, BT. So this is what is called as BT over there. Bacillus thuringiensis. To avoid this BT infection, we have BT cottons, BT brinjal. This is the Bacillus thuringiensis. This is how the Bacillus thuringiensis looks like. Group of bacteria is Streptococcus, all those round bacteria that you find. Staphylococcus, Serratia marcescens, and Bacillus thuringiensis. Source of infection over here, commonly in the summer and the rainy season due to humidity, hot and fluctuating weather. Starvation also leads to damage of the gut region, the digestive region, and leads to bacterial growth. The diseased dead larvae are the main source of contamination, because that is when the other larval forms, when they come in contact with this diseased larvae, the healthy larvae is also affected by that. So time and again, you're going to keep checking the condition of the larva, and if you find any infection, we are going to hand pick it up and you're going to burn them away. Some bacteria enter the body of the silkworm through mouth, that is consumption by contaminated leaf. And some bacteria are pathogenic when entered through the wound, and disease symptom appear within 2 to 3 days. So both ways, through mouth, that is by injection of infected in a bacteria, the mulberry leaf, or it can be due to wound, cut or a wound where it can easily pass through. Toxicosis is caused by consumption of Bacillus thuringiensis, and mortality is noticed in few hours, very toxic. So this is how the bacterial Flachery disease silkworm looks like over here. So you can see how dead and putrefied they are, completely flaccid, thin and flaccid. So this is how the bacteria is going to, the larval forms are going to look like by the infection of the bacterial Flachery. Symptoms. Flachery again means they are soft and flaccid. The disease symptoms are of general type, that is loss of appetite. They become lethargic, that is very tired. They don't move at all. The dead larvae becomes black, rots, and emanates foul smell. So this is how they are. They are black, they rot, and they emanate foul smell. Based on the site of the infection and pathogenicity caused by the bacteria, Flachery disease is called as. You call it as bacteremia when the bacterial disease is infecting the digestive organ, that is a cephalothoracic region, becomes translucent and develops diarrhea and rots with a foul smell. That is, they have those dysentery and they rot and die with a foul smell. So then you call them as bacteremia, that is disease affecting the digestive organs. You call them as septicemia, that is when the disease is infecting the hemolymph, that is the hemolymph is the blood of the larval worm. Greenish tinge of dorsothoracic region, soft blackened body, they rupture and rot with a foul smell. Dead larvae attains red color due to Serratia species. The Marcinensis infection is the one that causes that red color. So when the bacteria is infecting the hemolymph, you call them as septicemia. When the bacteria is infecting the digestive organ, you're going to call them as bacteremia. And you're going to call them as SOTO, that is when toxicosis is a sudden death. The symptoms of the larvae develops by paralysis, tremors, that is they shake a lot, they vomit, gut juice, hook-shaped body, they turn into a hook-like structures, and they rot fast with a foul smell. You call them as SOTO. So these are the three type of bacterial infection caused by the Flachery: bacteremia, septicemia, and SOTO, that is toxicosis. Disease prevention. So again, you're going to disinfect the whole room where you're going to rear them. Proper egg incubation should be done before development of embryo. You're going to avoid irregular molting and starvation. Care should be taken to accurately allow the worm to settle for molting. When they're molting, this larval forms needs lots of rest, and they don't want to be disturbed because the whole skin has to come out. Probably it might be a very painful procedure for them. The whole skin has to come out and they have to emerge out of that old skin into a new skin. So they need some place to settle and molt very calmly and peacefully. You're going to dust slaked lime to be applied compulsorily when the larvae are settling for molt. Slaked lime, why? Because they are going to absorb the moisture, if any, present in the leaves or even on the bed. The early resumed larva will not feed. That is, after molting, the larva will not immediately feed on the leaves, even though they have the available leaf over there. And in addition, lime brings down the humidity and they keep the bed dry and reduces chance of pathogenicity. You're going to provide requisite spacing, good ventilation, that is, you're going to avoid overcrowding. You're going to provide good nutritious mulberry leaf with good moisture content in them because they need some water. They don't drink water like what we drink. So whatever the moisture they are going to take is they get it from the good quality mulberry leaves. Leaf scarcity in the rearing bed due to drying, overcrowding, underfeeding causes starvation, which finally leads them to bacteremia flaceri. So these all have to be avoided. Those were the diseases of the mulberry. That is, we talked about the protozoans, we talked about the fungal, we talked about the viral, then we talked about the bacterial diseases over here. Now coming to pest, we talked about pest, that is the oozy fly and the beetles. This is what we are going to talk about over here, pest of mulberry. Under pest of mulberry, the first important pest is this oozy fly, Exorista bombycis, which is a major pest of silkworm. They usually look like common housefly, but they are a little bit bigger in size than the common housefly that we have at home. 5 to 15% of cocoon crop loss in traditional sericulture areas of South India. So 5 to 15% is definitely a major amount of loss of silkworm, given that the cost of the silk that we have over there. So oozy fly or Exorista bombycis, they come under Diptera family because they are the winged in flies. They are flies and they are winged. So all those winged flies, they are placed under Diptera. Trachinidae is the name that is given. The adult oozy fly is blackish gray in color. So you can see blackish gray in color. And it has 4 longitudinal black stripes on the dorsum. So you can see the longitudinal black stripes on the dorsum. And they have an abdominal crosswise stripes on the abdomen over here. So you can see this side, they have a crosswise on the abdomen, and they have that longitudinal black stripes on the dorsal region. So this is how we are going to differentiate between a common housefly and the oozy fly that you find, which is a pest for silkworm. The common name of the housefly is, of course, the Musca domestica. Oozy fly over there, mature maggot, causes reduction in the yield of cocoons and cocoon quality. What's a maggot? Maggot is nothing but a larval form of the insects. Here, for example, when you're talking about the oozy fly, the larval form of a oozy fly is called as a maggot. Okay. Cause of death of the silkworm larva because of this oozy fly. What are the symptoms? You can see over here, there's a black scar that is present on the larval form over here because of the deposition of the egg by the oozy fly. There's a dark scar-like structure. This is a oozy maggot, the larval form of the oozy fly over there. And this is a oozy pupa. Don't mistake them for the pupa of the silkworm. This is the pupa of the oozy fly, and this is the larva or the maggot of the oozy fly. And that is the egg of the oozy fly over there on the larva of the silkworm. So that small tiny dot that you find over there. Symptoms. Presence of creamy white oval egg on the skin. So this is an oval egg. Presence of white creamy color ones on the skin of the larva in the initial stages. Presence of black scar on the larval form. So you also find that black scar over there. Silkworm larvae die before they reach a spinning stage, so cocoon is not formed. So they are going to die before they are weaving the cocoon around them. And in a later stage, pierced cocoon is noticed. That is, pierced cocoon. When the cocoon is pierced, what happens? There is a loss of the silk thread, because even one thread being broken at one point, you don't have a continuous thread. And if you don't have a continuous thread of the silkworm, the value of the silk diminishes. So there will be a loss of economy also over there. So you have to be very careful to not be affected by this oozy fly, as the cocoons are being pierced and there's a hole in between them. So how they look like over here? So you can see the oozy flies laying eggs on the larval forms over here. And after the larval has been laid by the eggs, you can see the dark spots over here, something like the black pepper spots that you find. So you have those dark scars over there. And this is the egg that has been deposited by this oozy fly on the larval form. So this is a larva, the large larva, where you can find those magnified egg that you can see on the silkworm body. Yeah, so this is a egg laying ones over here, and again they have those black scars over there. In this picture, you can see this larval form where the maggot is emerging from the silkworm. Can you see from its body? Now you'll be wondering, how did the maggot enter a larval form? Is when the oozy fly laid its egg on the larva, the egg slowly moves inside in search of food. That is when the larva hatches out of the egg, they go inside, they pierce inside the host body for the nutrition to be taken away by them so that they can grow. And once they have grown to size and once they have to undergo pupation is when they pierce out of the host body. So this is emerging, that is, it is coming out of the skin, the body of the larva. So imagine this is the body of the larva and the maggot is coming from inside out. So emerging out from the silkworm. And not only the larval form, this is a cocoon of the silkworm over here. Even from the cocoon of the silkworm, you can see how the oozy maggot is emerging out. So if they're going to emerge out like this, they are going to definitely spoil the silk thread and they are not reeled properly. The silk thread, you cannot take them out at one stretch. The quality and of course the rate also falls down for such type of silk thread. So you have to be very careful in that. So here you can see how the oozy has pierced out of the cocoon. So these are all cocoons of the silkworm, and they pierced and they came out of this cocoon. So these are all are going to go waste for the sericulturists. So it's going to lead to some economic loss for them. Period of occurrence throughout the year, there is no particular season for them. Severity is more in winter season, of course. You have to maintain sanitary and hygienic condition in the rearing room, provide physical barriers like wire mesh in the doors and windows of the rearing room, something like what we keep a mesh for mosquitoes so that you avoid the mosquitoes that enter room or the door windows. You're going to spray 1% of benzoic acid over the larvae to kill the eggs of the oozy fly. And you're going to dissolve the oozicide tablets. So you have some oozicide tablets format. What you're going to do is you're going to take 2 tablets and you're going to mix it in 1 liter of water and you're going to put it in the tray, and that is going to attract the oozy flies into the tray, and ultimately they fall into the trap and they're going to die. So this is how you're going to trap the oozy fly by oozicide tablets. Okay. Now, why do you think they are attracted? Because those tablets contain something like a bait, the false bait, that the food is present over here. You can come and have the food over there. So once the fly comes and settles on that solution, they get killed. So that is how you are going to trap them. Okay, the next pest is the Dermestes beetle. So you can see the black beetle that you find here and there. This is an arthropod again. Most important species of Dermestes beetle is prevalent in egg production centers. Wherever the egg is produced, this particular beetles are present over there, called as Dermestes aether. Identification of the pest is the adult of Dermestes aether are black in color. So the adult is black in color, you can see over here. Elongate and oval in shape and about 7 mm in body length. That is almost about 0.7 centimeters in length. If you convert mm into centimeters, 0.7 mm. Grubs, you can see this white one is a grub over there. In oozy fly, the larval form was called as maggot. In the Dermestes beetle, the larval form is called as grubs. So don't get confused. It's only how you're going to differentiate between each species. Okay. So oozy fly, when you call them as maggot, you think that this is the larval form of the fly, and we're calling them as grubs. That is the larval form of Dermestes beetle. And the grubs are also black in color and are covered with bristles. So you can see over here, they are covered with bristles over there, the larval forms, and they form into a pupa. The next stage is the pupa stage. So you can see those bristles over there. These bristles are the one that is going to protect them from any injury. So these are the grubs of the beetle, Dermestes beetle, and this is the pupa of the Dermestes beetle. Okay, what is the symptoms of the pest attack and what is the extent of damage? The grubs are the main ones to cause damage. They infect the cocoons and they show the presence of multiple irregular holes on them. That is, they're going to spoil the cocoon completely. Though they show preference for feeding on the leftover or dead matter in the cocoons, they do attack some green cocoons also, as well as the egg-laying moth whenever their population gets increased. So it is an easy catch for them if the egg-laying moths are more in number. Estimated damage due to this beetle attack is, if it is in a pupae form, 16.62% of damage, and when the silkworm is in a moth form, 3.57% damage. And in the egg, there is a 20.919% reduction in the egg production in the grainages. That is where the seeds are being produced. Seeds means here the eggs of the silkworm. So this is the cost that will be affected for us because of the Dermestes beetle. Okay, so here you can see there are number of holes that have been made by this grubs over there. So here you can see all those grubs of the holes on the cocoon. So these are the silkworm cocoons. And here you can see how the moth have been killed by this particular Dermestes beetle, where you have those dark spots on them, and the abdomen is completely worn out, dead, and very hard type pose. So this is the caused by Dermestes beetle over there. Control measures mechanically, how you're going to control them. Collect the grubs and adult either by sweeping or by using a vacuum cleaner, and destroy them by burning or dipping them in a soap solution. Physically, exposure of the beetle-infested pierced stifled cocoon packed in HDP packets or the bags that you have to sunlight for a period of 6 hours. This will also remove the beetle infection. Chemical, what are you going to use for as a chemical? Wooden articles of the cocoon storage rooms and grainages should be dipped in 0.2% malathion solution for 2 to 3 minutes. Here, when we are talking about 0.2% malathion, means we are going to use only 0.2%. That is, if it is in a solution form, you're going to take 0.2 ml of malathion, mix it in 100 ml of water, and you're going to dip all this cocoon in that particular. You're not going to use more than that. Say, if you're going to use 0.5% malathion, that is not recommended. You're going to use excess of pesticides over here. And what you're going to do is, after dipping them, what is that you're going to do with that toxic, the pesticide? You're going to throw them in the soil. That soil is being infected, is contaminated by this toxicity. And this is getting washed out in the river water or pond water during rainy season. The water is also contaminated by this pesticide. Ultimately, all the living organisms at one stage or the other, they will be affected. So in toxicology, when they mention this particular amount to be mixed, you're going to mix only that particular amount. You're not going to increase anything more, or you're not going to use lesser amount. So that is the importance when we say 0.2%, 0.5%. Percentage means 100 ml. In 100 ml, how much you're going to add? That is 0.2 ml in 100 ml of water. You're going to prepare that malathion. And after 10 days, the tray should be thoroughly washed in water and sun-dried for 2 to 3 days, because you're going to remove any infection that has been present. You're going to store the pierced cocoon in deltametrin treated bags, that is at 0.028% deltametrin. This is even more lesser, 0.028% of deltametrin you're going to use for controlling this particular grubs. Okay, prevention. Always clean, disinfect the room. Storage of the rejected cocoon and perished eggs in the grainages for long period should be avoided. You are not going to store this infected one, because once they are infected, they have to be immediately burnt. Otherwise, they are going to infect the other healthy larval forms. The cocoon storage room should be cleaned thoroughly. Before and after emergence of a silk moth, the grainage premises should be kept clean and tidy. Provide wire mesh to the doors and windows so that you don't allow these beetles to enter inside. And they have to be free from beetles and grubs. So you're going to store them in a neat room. So that is the preventive measure of this particular. So that was about the diseases of the silkworm. Now we'll be talking a little bit about how we are going to rear them. That is how you're going to take care of this larval forms, how you're going to take care of the egg, how you're going to take care of the silk moth for production of the eggs. That is called rearing. Reeling is a reeling technology. Here, you're not going to talk them as culture. Under reeling, you're going to talk them as technology, because reeling means you're reeling that silk thread into a reel, the bundle of thread of bundles, which is again sent for fabric production. Okay, so for that you need a raw material, that is the thread. So you're going to remove the thread from the cocoon. That is called reeling technology. So you're going to talk in two topics of your rearing and reeling technology. You can see over here such wonderful moth of different silk. Here you are going to talk about mulberry. Then the yellow one is a tussar silk. Usually they are yellow. The threads are yellow in color. The muga silk and the eri silk moth. So by seeing this moth shape, you should be able to tell what type of silk you're going to get. Okay. What are the stages? That's what I was talking about. You're going to talk about them as 3 stages. Cultivation of mulberry comes under agriculture division. Why agriculture division over here? Because you're going to grow these plants, the bushes, mulberry plants you're going to grow, and you're going to take care. We talked about this cultivation of mulberry in the previous online class over there. You can go and have a look at how the cultivation of mulberry is. So that because you're growing plants, so they are called as agriculture division. Rearing of the cocoon, entomological division. Because you're going to talk about the insects, you're going to grow the insects. So they come under entomology. Insects means entomology. So rearing of the cocoons, you're going to rear the egg to larva to pupa, the cocoon formation, and then to moth. So they come under entomological division. Reeling of the cocoons, I told you, you're removing the thread from the cocoon. That is technical division. Technology comes over here, where you're going to use some machines to remove the thread. So that is why they come under technical division. So you'll be talking, sericulture means 3 divisions. You can get into any division you want to. Say, for example, if you are thinking of planning a small scale industry where you want to rear this sericulture, you can take up any division of sericulture, or you can do everything at one roof: agriculture, entomology, and technical division. So before going into the technical, entomological and technical division, we are not going to talk about agriculture division because we have already talked about it in the previous classes. We're going to talk only about entomological division and technical division over here. So before going on to that, we are going to talk a little bit of silk secretions, though I had already talked about it in the morphology of the silkworm over there, a biology of the silkworm. You have to understand that silk secretion is of 2 types. It is a fibroin. One protein is a fibroin protein. Another protein is a sericin protein over here. The fibroin makes 60 to 70%, while sericin makes up to 25%. Fibroin is tough. What is the quality of a fibroin? I talked about this in the previous class again. It's going to be tough. It's going to be elastic, and it is insoluble. You cannot solubilize them. They make the core of the filament in the form of brins. The thick thread is because of this fibroin. So they have that thickness. They are very tough, and they are elastic. So that is the reason why they are used in parachute making. They're also used as a fabric for military personnel. And these are also used in medical purpose, where I talked about these threads are being used as sutures to stitch any cut open part of the body during operation. So because they are very tough and elastic. And also one other advantage is we don't have to remove the threads again. And after some period of time, they can become insoluble because they are in the digestive juices inside the body. Okay, sericin. Sericin forms 25%. They form the cover around the fibroin. They just form a cover, gelatinous cover, to take care of the fibroin over there. So that's the gelatinous or a gummy portion of the silk. They are easily soluble, hot water soluble protein, and holds the brim together. That is, the fibroin is holding. So you can see over here, so this is a fibrin, fibroin. protein, and sericin is the one that is covering them around, giving a protection like layer around this fibroin, though they don't need any protection. But when they are wound, as in a cocoon formation, they form a protection around the fibroin. So when you're talking about them in weight in grams of 900-meter long silk filament, you're calling them, the grams is converted as deniers over here. So denier is, the size of a normal cocoon is 1.8 to 3 deniers. That is 3 grams, you call them as. Sometimes they call it grams, sometimes you can also mention it as denier over there. So a single cocoon weight is 1.8 to 2 or 3 grams, and its shell, without the enclosed pupa, it's only 0.45 grams. So when the pupa is removed out, their weight becomes only 0.45 grams. But along with the pupa, you can have it anywhere from 0.8 to 3 grams or 2 grams. And you need 2,500 cocoons, that is 2,500 cocoons are needed to yield just 450 grams of silk. So you can imagine why the silk fabric is so costly, because this much of cocoon is necessary, 2,500 cocoons to yield approximately half a kg of silk, right? So that explains why they are very costly. And of course, because of their tough elasticity and long-term investment, you invest once and it stays with us for the whole of the life, right? You might also have been seeing that even your grandmothers will be handing over their silk fabric, that is usually the silk saris, is handed over from generation to generation. The silk clothings of the kings and the queens is still preserved. There is no damage at all. That explains how turgid or how tough and how important is silk, and you can keep them for long period of time. So that is the importance of the silk over here. Coming to rearing, how we are going to rear them, that is how we are going to culture them, is called as rearing of silkworm. So first thing is you're going to select the silkworm race over here. You have 3 types of races: univoltine, bivoltine, multivoltine. What is this univoltine, bivoltine, and multivoltine? Univoltine is single. They're going to have a life cycle only once in a year. One complete life cycle once in a year is univoltine. Bivoltine, in one life cycle, they can produce 2 generations. So that is called as, that is in 1 year, you have 2 life cycles, is called as bi. Bi means 2. In 1 year, you have 2 life cycles, bivoltine. And in a multivoltine, you can have 5 to 6 life cycles in 1 year. What is the difference? In 1 year, if you're going to have one single life cycle, you're going to produce less amount of cocoon. In 1 year, if your silkworm race is going to have a life cycle of 5 to 6 life cycles, then you will have more number of silk cocoons forming, more number of silk thread. Again, it depends on the quality. Why? Quality over here, because you are forcing the moths to keep producing cycle by cycle. We are not giving time for the larval form to grow properly. If you're going to give less time for the larval form to grow, the protein content also is very less. If you're going to give more time for the larval form to grow, you're giving more proteins for them, and you have high quality silk produced from that univoltine. Of course, it all depends on the type of the silkworm race that you are going to rear them. And silkworm seed production. What is this? Hybrids of multivoltine and bivoltine are reared commercially for its superior quality and huge quantity of eggs. So you're going to have a hybrid varieties over here between multivoltine and a bivoltine for a proper superior quality eggs, so that you get superior quality silk. Grainages. What is grainages over here? Where you have those seed production. Seed production over here means eggs of the silkworm. So these are the places where the seeds are produced at large scales. Healthy, disease-free cocoons of pure race are selected, preserved at 23 to 25 degrees centigrade in wooden trays at 70 to 80 percentage of humidity. After emergence, that is after the emergence of the moth from the cocoon, the female moth are transferred to plastic trays, allowed to copulate with a male of desired race under a black cloth for 3 hours, and within 12 hours, you have 400 to 600 eggs within 12 hours after the emergence of the silk moth. Here, we also discussed about how you are going to separate a female moth from a male moth. We also saw the sexual dimorphism, where you can clearly demarcate between the male and a female. That is the antennae, larger antennae in the male, smaller antennae in the female, narrow body of the male, broader body of the female. And then also the abdominal side, you have some dimorphism or demarcation that you can separate them. So here you have 5 stages of rearing. 5 stages of rearing. What are the 5 stages? First stage is supply of eggs. First, you need supply of eggs. See, I'm going to start a business of rearing silkworm. How do I start if I don't have eggs? So I'm going to procure the eggs first. So the supply of egg is needed from a particular place where only eggs are being reared and sent or supplied to different organizations. So quality of the seeds is important first. You can't just take any type of eggs, because this can be infected, then your time, money, everything goes waste. And only those eggs are given that that has been produced in a well-recognized grainage. So you're going to select the eggs only from a well-recognized government-run ones, because they are the ones that are going to take care of proper health of the eggs. Incubation of the eggs. So how we are going to incubate them? Incubation condition should be optimum for univoltine. Univoltine, only one life cycle per year. 73.5 to 77 degrees Fahrenheit is the optimum temperature you're going to give them to incubate the eggs. That is the heat. You're going to give the heat for the development of the egg inside the eggshell. And for bivoltine, you're going to give 80 degrees Fahrenheit, and for for 77 degrees for bivoltine and 80 degrees Fahrenheit for multivoltine. So these are the prescribed temperature for incubation of the eggs. And that is the second stage. And for incubation seeds, they are spread on a piece of a paper in a rearing tray. So what you're going to do for incubation, you're going to spread a paper and you're going to place those seeds and incubate them in a piece of a paper. This is the second one. On the third one, brushing of worms. What do you mean by brushing? You're not going to brush the teeth over there. If you can see over here, so can you see this tray? This is a paper where they had incubated the eggs. After that incubation, when the eggs are being hatched, you will find this tiny, small silkworms. They look like ants over there. Very small ants over there, and you have this eggs that are being produced over there. Hatched ones are something like this. Once they are hatched, they are being brushed with a feather of a bird. So this is a feather of a bird. You're not going to use a painting brush for brushing them or separating them. You're going to because they are going to be very tiny, small ant-like structures. So you're going to be very careful in brushing them, slowly removing them and putting them in another tray. So that is brushing of the worms. Rearing of the worms. Rearing means you're going to take care by giving them proper food. What is a proper food for the larval forms? You are going to give them mulberry. This mulberry in the first stage, that after immediately they have hatched, they need very tiny leaves. So what you're going to do, you can't give that large leaf for them to feed on. You're going to cut them, chop them into small pieces, and you're going to spread them on a tray, and you're going to release, brush all those worms that have been hatched newly, and you're going to put it on this tray, and they're going to feed. That is, you can see over here. So this is the first feeding mechanism, and all the silkworms are put together in that particular tray. And cleaning, of course, you have to time and again take care of the cleaning. You're going to take care of the spacing. Then you're going to take care of the mounting. This cleaning spacing is all the different instar larva. That is, after each molting of the larva, you're going to remove that larva and you're going to place into another fresh bed where you're going to give them with a new fresh cut leaves. So here, when it is very difficult to touch the larva and put them into another tray. So what you're going to do is you're going to place this larva beside the new freshly cut leaves. All these larvae slowly they crawl to the new fresh leaves. Then you can clean this aside. So that is how you're going to clean the trays. And of course, spacing, you're going to take care of the spacing because as the larva is growing after each molt, they become big. When they're becoming big, you cannot put everything in that same tray. So you're going to make space so that they can grow, they can eat properly. And here you can see over here, this is a second instar larva silkworm where you can keep them in single tray. But the same one, when they go into a fourth or a fifth instar larva, what you're going to do is you're going to make them separated into 4 different trays because they're growing in size. And you can see the fifth instar larva, they're feeding even on the shoots of the plant because they are very hungry. And as we talked about them, they are very voracious feeders. They just eat, and eat. Okay, the next stage is mounting and harvesting. What do you mean by mounting? You're going to mount the fully grown larval form onto different trays so that they can settle at one place. They can form cocoon around them. That is, they spin out the silk and they form a cocoon and they pupate in that particular way. So this is the fifth, the last stage, the spinning of the cocoon. So what is happening over here? This worm, the larval forms are handpicked. And after the handpick, they are put into some different places where they can form the cocoons over there. So within 3 days, they start to spin. How do you find out that they are ready to spin? Is when they stop feeding. They stay stationary. They do not move. They do not eat. Is when you will identify that they are ready to form the cocoon. So this is the stage when we are going to handpick them. So this is how they are handpicking them, and this is how they are mounted on a mountain edge. So you can see those small boxes. This is a particular small tray where you have those demarcations. So each larva is going to take up one particular box over there. They're going to find out which one is suitable for them. It is their wish wherever they want to go. So they are going to settle down in these small boxes and they're going to spin that silk gland around them. So here, the silk gland comes into play. The spinnerts that we found on the mouth of the silkworm, they're going to spit out that silk gland and they're going to form that silk thread around that cocoon. So here you can see over here. Yeah, see. So you must have also seen sometimes at home also at some corners, this larval forms, they form that cocoon. So the next time they are going to form a cocoon, you can have a look at them, how the head keeps moving from one side to another so that they are forming that silk gland is being spun around them. This is a partially spun cocoon. So this is a mountain edge where we talked about. So each larvae is going to select its own box and they're going to form the cocoon over there. And this is a fully spun cocoon on a rotary mountain edge. This is where they keep on rotating them completely. And these are the different types of mountain edges. There are not just 1 or 2 type of mountain edges over there. So here you can see this is a traditional bamboo mountain edge. You have a plastic collapsible mountain edges. That is that we put it around the plant for protection. So that's a plastic collapsible mountain edge, a rotatory mountain edge. This is something like a bottle brush where each cocoon can just go and take its place and they can spin. This type of ones are very easy to collect them. This is the twigs where the twigs are also used for cocoon formation. And of course, this is a modified hybrid mountain edges where you have those box-like structures, each one settling in one single box and they form the cocoon. So these are the types of mountain edges. Mostly in India, you have this traditional bamboo mountain edge where they're made out of the bamboo.
Excuse me, ma'am. Important questions,
it's easy for the larval forms to stick and form that cocoon over there. Okay, quality of the cocoon, it depends on the raw silk filament. It also depends on the type of the leaf that you have given them. We are running short of time. Anyways, I'll try to finish this and leave you all. So Marketing of the cocoon is one more important one, because fixed price is given for this cocoon. The businessmen, they cannot give any type of amount, or they can't ask any amount for it. So you have a fixed price over there. And post-cocoon processing, that is a method of obtaining silk from the cocoon, is called as post-cooking cocoon formation, is where you're going to talk about the reeling technology. Now, till cocoon formation, you're calling them as entomological culture development. Now you're going to talk about the reeling technology, where it you're going to talk a little more about the machines that are used to remove the thread former reels. So this processing is called stifling and reeling. Stifling is killing the cocoon. Why? It is necessary to kill the cocoon, though it is very hard to say that, but yes, you have to kill the cocoon by placing them in a hot water, or you're going to boil them. Why? Because you're going to kill the pupa inside that cocoon. Why you're going to kill the pupa inside the cocoon is because once the pupa starts to emerging out, it's going to tear the cocoon. If it's going to tear the cocoon, as I told you, you're going to rupture the silk thread. So if you're going to rupture the silk thread, that becomes waste, or they don't have a proper value of that silk. So you're going to kill them, and then you're going to reel them out. So that is called as stifling. Stifling means killing of the cocoon. And 8 to 10 days cocoon is selected. They're dropped in the hot water, exposed to sun for 3 days. This is how they're being harvested. First, harvesting is they are going to be collected from this cocoons formation. After collection, they are being put in the baskets. And these are defloss. Defloss is any extra hair, the silk that is present, they are removed, and the proper cocoon is kept over there. Cocoon stifling. So here you can see how they are going to kill the pupa in the cocoon. To avoid the moth emergence, to protect the quality of the cocoon, the silk, to preserve the cocoon for reeling, and dried cocoons can be stored for a longer period of time without purification or without any mold formation, petrification, the death of the silk. Stifling method, there are different types of stifling methods. Sun drying is one type. Steam stifling is another type. Steam stifling is you're going to steam them in a basket, or also you can steam them in a chamber steaming. So here you can see all the cocoons are kept in a basket, and they're steamed in a hot water vessel. And sometimes you can also use a hot air drying. Hot air drying, you can use them in a hot oven. Just put it in a hot air oven, and they are killed over here. So here, sun drying is one type. Barrel steaming is one type. This is a basket steaming, where the vessel is kept. The cocoons are kept in a basket and covered with a cloth. Basket steaming. This is a chamber steaming, where you have a box where the heat is given to the chamber, and that is being dried over there. This is a hot air drying chamber on a large scale, where they can be used. And this is a hot air drying chamber, something almost like that of the what you call them as oven type of thing that is kept over there. Okay. So cooking is one over here. So you have pan cooking, you have 3-pan cooking, you have pressurized cooking, boiling point over here. So they are used over there. And silk reeling, the last topic is silk reeling. How the thread is being spun is what we are going to talk about in the silk reeling. Technical division is what this is formed over there. This involves the last step of sericulture, energy consuming. 30 to 40% of the cocoon is sent for pupae formation. That is, pupa is stored because you want those moth to be cultured for egg production, and the rest is gone for producing silk fibers. And this is a post-cocoon process, and 1 kg silk needs around 5,500 to 6,000 cocoons are needed for this purpose. Okay. And reeling process over here. What is a reeling process over here? They said that time is over, but anyways, let me continue. Aisha, are you there? Okay, so let me
Yes,
continue.
ma'am. Yes, ma'am.
You have any doubts, Aisha, till now?
Important questions, ma'am.
Aisha Begum?
Important question answers, ma'am.
Okay, if you're listening, then let me continue this. Reeling processes where the threads are being removed. So this is the machine that are used to remove the thread over there. And this is a chakra that is used for removing the threads. This is what you can see over there. And this thread is woven as a thread in the chakra over there. If you can remember the Gandhi chakra where they take out cotton thread over there. These are some of the machines that are used for cotton reeling method. And these are the steps that you find, how they are being processed. These are different steps over there. And this is how the weaving is done. The silk weaving is done. And what are the uses over here? These uses are where you have, you can form them as silk cloths, synthetic fibers, military purpose, fishing fibers, parachutes. You also have tires for racing cars, insulation of the telephones, and medical dresses, that is suture making of this particular one. Okay, so this is what is all about diseases of sericulture. Any doubts, ma? If you have any doubts, you can just ask me.
No, ma'am. Madam, important questions.
If you don't have any doubt,
ma'am, pass marks how, ma'am?
is that
for last
okay with you?
Pass marks, ma'am.
Then we'll try to wind up the class. Thank you for listening to my class.
Madam, pass marks how much, ma'am?